Hello students, welcome you all to a new video of Sunanda's tutorial. So today we will discuss on the topic life cycle of Selaginella. So Selaginella is characterized by an alternation of generation alternating between a dominant diploid sporophyte. Obviously all the pteridophyta have a dominant phase, dominant diploid sporophytic phase, and a very little smaller haploid gametophytic phase. It is a key evolutionary plant because it exhibits heterospory. That means Selaginella can produce two different types of spores. It is a very crucial step towards seed habit. So it is a major evolutionary step or development which was observed in Selaginella for the first time, that is the heterospory. Actually two types of phenomenon are there. One is homospory, one is heterospory. Some pteridophytes show homospory, that means they can produce only one type of spores which are morphologically as well as physiologically same or are of same type. But heterospory species can produce two different types of spores which are morphologically as well as physiologically different. Okay, next. At first we will discuss about the sporophytic phase of Selaginella. The dominant plant body is sporophyte with true root, stem and leaves. Strobilus, these are present at the tip of branch with specialized leaves called as sporophylls, and these are compact cone like structure called strobilus, and many strobilus consist strobili. Spore production. The strobilus contains two types of sporangia, that means spore sac. Microsporangia, which produce microspore. Megasporangia produce megaspores. Okay. So this is the Selaginella plant. This is the stem or axis. These are small leaves. This is root and this is rhizophore. Okay. Then stem anatomy. The cross section of stem shows one epidermis layer with a thick cuticle which is protective in nature to prevent the water loss. Then cortex layer is there. Under the epidermis, cortex layer is there with hypodermis, then inner cortex. In younger or green stem, these inner cortical cells contains chloroplast for food synthesis. So inner cortex, some cells of inner cortex in younger stems, in younger plants of Selaginella have chloroplast for photosynthesis. Then air cavity and trabeculae. Air chamber, a large distinct air gap physically separates the inner boundary of cortex from the central vascular cylinder. So the central vascular cylinder is separated from the cortex by some air chambers, and that central stele is held in place across this air chamber by radially elongated filaments called as trabeculae. Okay, these trabeculae are modified endodermal cell. Actually the inner layer of cortex is called as endodermis, but here that endodermal layer is modified into trabeculae. Okay, and can be identified by the presence of Casparian strips or Casparian bands. Okay, then stellar part, stellar region or stele. So it possesses protostele where the core is completely solid and entirely lacks the central pith. So it shows protostellic condition where the pith is completely absent. Pericycle, the vascular tissues are tightly enclosed on the outside by single layer of pericycle cells. Then xylem, positioned at very core, and xylem is typically exarch and diarch. Okay, exarch means protoxylem is placed towards the outside and metaxylem towards the inner side, and diarch means typically two vascular bundles are there. Then phloem, it is a continuous layer of phloem completely encircles and enclosing the core xylem, making the arrangement amphicribal. So this type of arrangement is called amphicribal arrangement. Okay, next. So it is the internal structure of stem. These are the trabeculae, these are air spaces. Okay, and this is the anatomy of Selaginella stem. Okay, now the anatomy of leaf, which shows upper and lower epidermis and mesophyll tissues in between upper and lower epidermis. Intercellular spaces are there with central vascular bundle. So the vertical section of Selaginella leaf reveals three primary anatomical zones. One is upper and lower epidermis with a thick cuticle. Stomatal pores are mostly restricted to the lower epidermis. So here it shows hypostomatic condition. Chloroplast, which are uniquely present in epidermal cells. Okay, mesophyll, these are ground tissues located between upper and lower epidermal cell. Okay, then vascular bundle present at the in the middle, and bundle sheath, which is the protective layer, which can be sclerenchymatous or parenchymatous in nature, enclosing the mid vein. Xylem is located centrally, consisting of tracheids, which helps to conduct water, and phloem completely surrounding the xylem. It also shows the amphicribal arrangement. Okay. Next, the ligule. Ligule is present at the base. Okay, and that ligule is can be differentiated into a elongated part called ligule and a broad base called glossopodium. Okay, glossopodium is the submerged semicircular base of ligule made up of large empty cells. Okay, and glossopodial sheath, which is distinct ring of tubular cells enclosing the glossopodium. Okay, and the function of ligule is it can act as water secreting or water absorbing organ, keeping the young apical bud moist. Okay, so this ligule is placed here on the adaxial side of the leaf, Selaginella leaf, and the anatomical feature shows this ligule have an elongated part and a broad base called glossopodium. Okay, then anatomy of root. The cross section of root reveals concentric tissue layers tailored for nutrient absorption and transportation. The outer layer is called epiblemma or epidermis, enclosing cortex. Then inner to cortex endodermal layer is there with barrel shaped cells and Casparian strip. Then inner to endodermis pericycle is there. Then vascular bundle, which is organized in radial pattern. That means alternating patches of xylem and phloem is there. Xylem development is exarch. That means newly formed xylem takes place towards outside. Newly formed xylems are placed towards outside, and matured xylem or metaxylem is placed towards inner side. Okay, next. So it is the transverse section of Selaginella root. Then anatomy of rhizophore. Rhizophore is an organ peculiar to Selaginella that grows downwards from stem branch, points to produce true root when it hits the ground. So it originates from the stem and grows towards the soil or ground, and after touching the ground they produce true root. Its internal anatomy sits halfway between a stem and root, we can't say it is a true stem or true root. The outer layer of this rhizophore is epidermis. Next, inner to epidermis, cortex is there, which can be divided into outer and inner cortex. Then inner to cortex, endodermis and pericycle layer is there, and the vascular bundle is mainly protostele. Okay, so it is the transverse section of rhizophore of Selaginella species. So after this, now we can proceed towards the gametophytic phase of Selaginella. So that strobili produce microspores and megaspores inside microsporangia and megasporangia respectively. So those spore mother cells after meiosis produce megaspores and microspores. So megaspore mother cell produce megaspores after meiosis. Microspore mother cell after meiosis produce haploid microspores respectively. These spores after release into the environment can germinate independently to form gametophytic generation. Microspore germinate to form male gametophyte and megaspore germinate to produce female gametophyte. Microspore germinate inside microsporangium to produce biflagellate sperms or anthrazoid. Megaspore develop into multicellular female gametophyte which protrude out from the megaspore wall and develop female sex organ containing egg, and that female sex organ is called archegonia. Okay. Then water is very essential for fertilization. Motile anthrazoids or sperms swim through the water to reach the egg cell which is placed inside the archegonia. Then after fertilization they form diploid zygote which develop to make a new sporophytic generation. Zygote at first develop into embryo, and that development relies or depends on the female gametophyte for initial nutrition. Eventually it grows into an independent matured plant completing the whole life cycle. Okay, next. So this is the diagrammatic or graphic representation of the whole life cycle of Selaginella. It is the sporophytic plant. They produce micro and megasporangium. Then after meiosis, microspores and megaspores are formed. Then from micro and megaspores, male and female gametophyte produced. Then fertilization, inside gametophytic plant, archegonium and antheridium are produced. Then inside the archegonium egg cell is formed, and inside the antheridium many anthrazoids are formed. Then after fertilization zygote is formed. Zygote germinate to produce the sporophytic plant. So with this the whole life cycle of Selaginella is completed, and I am concluding my lecture video here. I hope you will get benefit from this video, and if you liked it, please hit the like button, share with your other friends, and please subscribe to my channel Sunanda's Tutorial. Thank you.